All right, can you guys all hear me in the back? Great. So first of all, thanks, Mark. Really appreciate being included in this group. It's been a fantastic day of science, and I'm really excited to tell you guys about the work that we've been doing looking at functional specialization in the mouse visual cortex. So to me, one of the most amazing things about the central nervous system is the ability to take incredibly simple biophysical inputs at the periphery and transform them into the complex representations and perceptions that we experience every day. And I think that the visual system is a particularly good example of this, where light hits the photoreceptors in the retina, and this information is then transformed into electrical signals that are propagated from the retina to the thalamus and to the cortex in parallel streams where it's segregated and transformed into increasingly complex representations. Such as, for instance, in area MT, which is well known for having a very precise representation of the speed and direction of stimuli, or the inferior temporal cortex, which has exquisite representation of very fine colors or of faces. And these transformations have really captured the imaginations of neuroscientists for many years, and many models exist about how these kinds of transformations could take place through the convergence of different anatomical inputs and through physiological transformations that might take place due to recruitment of inhibition and excitation. However, to get at this level of trying to really understand the synaptic mechanisms that might underlie these models, what I and many others have done is resorted to moving to the mouse visual system to try to use it as a model to understand visual processing. And while this has become a very popular activity in the last 5 years, and maybe don't need to justify it quite so much anymore, I do want to take a moment to step back and think about whether the mouse is a reasonable system to be studying visual processing. It has these tiny little beady eyes that are on the side of its head. It doesn't really have much of a binocular zone. It doesn't have a fovea, and so its visual acuity is very low. And it's really just not very much of a visual creature. Most of its default senses seem to be olfaction and somatosensation. But I think that but we need to really understand whether we can maybe train these mice to use vision as a mode of sensation, and whether we can then use that behavioral tool to understand how well they can see and what kind of transformations do take place to be able to study visual processing in this system. And so my lab has been pushing hard on different behavioral tools for the mouse. And I want to share one of the tasks that we've developed most recently, which is a two-alternative force-choice contrast detection task. And in this task, the mouse's job is to use this treadmill here to indicate whether it sees a stimulus on the left or the right side. And so it slides it in the appropriate direction. So here's the mouse performing this task. There's a tone where the trial starts. And you can see that he slides the treadmill to the left to signal a stimulus on the right. And that's because the way that we've trained the animal to do this task is by tethering the motion of the treadmill to the motion of the stimulus on the screen. You can see that flash there on the correct tone. That's another tool that we use to really try to engage the animal with that visual stimulus. That's a combination of the tethering of the treadmill and that stimulus on the screen. We hope really gets the animal to use the visual stimulus. So you can see there, he got it most of the way to where the threshold is at the center of the screen. His job is to move it to the center. And then it didn't get quite all the way, and so we had to futz around with it to get it there. So this is an animal that's partly trained, so it's still in the training process. Once animals become proficient at this task, we can uncouple the motion of the treadmill to the motion of the stimulus, and the animals will continue to do this task where they simply then respond to the left or the right according to the position on the screen. And then we can change the contrast of the stimulus to look at the animal's sensitivity. And so here's an example session from a mouse. So this is a single session in which a particular mouse did about 250 trials. They'll range between 200 and 400 trials a day. And what I'm plotting here is on the x-axis is the contrast difference, the right minus the left. And so for a contrast of 1, what that means is that since the stimulus is always on one side, that the contrast is on the right. And since on the y-axis we're plotting the percent of right responses, it means that 100% of the time that the stimulus is on the right, he goes to the right. And conversely, if the stimulus is at -1, that means it's on the left. And so 0% of the time, if it's on the left, does he go to the right. And if we then present a range of contrasts in between, where positive ones are stimuli of low contrast on the right and negative ones are stimuli of low contrast on the left, he has more variable behavior. And so sometimes he gets it right and sometimes he gets it wrong. We can then look at the reaction times during this task. So this is all of a cumulative distribution of all of the reaction times for this mouse. We constrain it that they can't respond until about 850 milliseconds after the stimulus appears. But even still, the animals respond with about 90% of the time within 2 seconds. They have up to about 25 seconds for each trial before we end the trial and start a new one. And importantly here, the green is for right trials and blue is for left trials. And that generally there we can train them so that they have very little bias in reaction time between the two sides. We can be more quantitative about this reaction time by plotting the average response time in terms of the contrast difference. And so what's important here is that the animals have faster response times for easier stimuli and slower ones for harder stimuli. And this is really consistent with measurements in humans and non-human primates that as the task gets more difficult, reaction times get slower, maybe as a suggestion of changes in the animal's confidence. And finally, we can take this data and use it to measure the animal's threshold. So here now I'm measuring the contrast difference in absolute terms, where 1 is 100% contrast on either the left or the right. And then importantly, the animal works at 100% contrast, 100% correct when, for these easy trials, and that falls off with contrast, and we can measure a threshold of around 10%. And so I want to point out that while 10% might not seem like a very sensitive contrast threshold, this is actually fairly comparable to what contrast detection you might have in your periphery, where the density of photoreceptors is actually more comparable to the density of photoreceptors in the mouse retina. So this is a summary of 4 different mice that we've trained on this task, and across multiple sessions where the error bars are confidence intervals. And I just want to really point out that these mice are extremely good at this task, that for all of them, they always respond on the right when the stimulus is on the right and always respond on the left when the stimulus is on the left, and that they're relatively robust to failure, and so we can probe them at their threshold level and they'll continue to perform at high levels. So one thing that we want to know, I think, and similar to what Carl told us about earlier today, with these simple tasks, we want to understand cortical transformations potentially. Is the mouse actually using the cortex to perform this task? And so in order to get at this question, we took advantage of a line of mice that express Cre recombinase in their albumin-positive interneurons, which are a population of interneurons that specifically target the cell bodies of neighboring principal cells. And so if we infect these neurons with channelrhodopsin using a virus, then we can activate these cells on interleaved trials and thereby determine whether, if we pair the activation with the presentation of the stimulus, whether it affects their behavior on this task. And importantly, we put the channelrhodopsin specifically in their left visual cortex, which controls their responses to the right visual field. And so our expectation is that activation of these neurons will specifically affect their ability to detect stimuli on the right side, but not on the left. And indeed, that's exactly what we see. So again, plotting the contrast difference of right minus left on the x-axis. And this is a single mouse, but over a number of sessions, what we see is that under control conditions, and these are interleaved trials, we have a really nice symmetric psychometric function. But if we then activate pyroalbumin-positive interneurons, we specifically have an impairment for stimuli on the right visual hemifield. And this is really important because it suggests that this isn't simply an effect on the animal's arousal, that we're not startling him with this activation. There isn't these kinds of cognitive changes. This is something that is really localized to those neurons that have the receptive fields for this visual stimulus. So I just wanted to show you that data to suggest that mouse vision is in fact sensitive, if not highly having high acuity. They can perform visual tasks and be very good psychophysical subjects, that they can work at 100% correct if they're well trained. And the mouse visual cortex does seem to be important for visual behaviors. However, this is really only half the hurdle of trying to determine whether the mouse visual system is a good model for studying visual processing. The other half really is a question about whether the visual system has analogies to other more complex systems. For instance, whether there is parallel processing of information in the visual cortex. Are there hierarchical transformations that are made? And is there functional specialization in these higher areas that would be akin to, if not analogous to, or not homologous to, rather, area MT or the inferior temporal cortex? And so I think that this image here, to me, is one of the clearest examples that there is parallel processing in the mouse visual cortex. So this is an experiment from Andreas Berghalter that he did, in which he made 3 different dextrans injections into the different retinotopic locations within primary visual cortex. And they made a tangential section through the brain in order to reveal the anterogradely labeled projections of these neurons to the higher visual areas. And so what you can see is that this population of neurons in V1 directly projects to and innervates an array of different higher visual areas. Importantly, the strongest projections are to these 3 areas, LM, AL, and PM, which are poorly named acronyms for their locations in space. But that it does have projections to an array of other areas, and some that aren't even shown here. A second thing I'd like to point out is that these projections maintain the retinotopic organization that was set up in primary visual cortex, and that each of these higher visual areas has a complete, if not equally balanced representation of the entire visual field. And to me, this really suggests that each of these areas probably has a unique representation of the visual world and might be important for processing specialized features. Andreas Berghalter then went on to look at the connectivity between each of these areas. And so here the thickness of the line is representative of the strength of connections between a given area. And while this looks like a hodgepodge of connectivity where it's kind of an all-to-all connection, what he demonstrated is that there actually is segregation of these networks into functional streams similar to what we see in the primate system. That he identified a ventral stream that these areas tend to project to more perceptual or sensory-related targets, such as the superficial superior colliculus, the amygdala, or the entorhinal cortex, whereas these more dorsal motor-related areas tended to project to the motor cortex, the cingulate cortex, or the deeper layers of the superior colliculus. So this really sets up a nice anatomical structure that suggests that there is parallel processing and functional specialization within the network. But we still really need to know whether these areas are all actually functionally specialized. And so in order to address this question, when I was in Clay Reed's lab, we developed this preparation for visualizing the entire mouse visual cortex. And so we implant a 5 millimeter cranial window over the dorsal aspect of the brain. And this reveals primary visual cortex as well as the array of different higher visual areas it projects to, along with potentially some somatosensory cortex and other cortical areas. So we can then use these windows to be able to identify functionally the location of the different higher visual areas within the window. And so we do this by mapping it out with intrinsic signal imaging. And so here, decreases in fluorescence correspond to increases in activity. And so you can see that as we move a visual stimulus to different places within the visual field, so from medial to lateral, the representation in these areas move to different cortical positions. So V1 here moves from lateral to medial cortex, as you might expect here from this mapping that Andreas did. And similarly, you can see mirror reflections of LM and AL, which are separating from V1 as you move into these more lateral positions. So we can use these maps to target areas for expression of genetically encoded calcium indicators to look at the activity of individual neurons within these areas. So this is an example experiment of 2-photon imaging of a single neuron and its responses to a range of different spatial and temporal frequencies. So here's an example of the types of stimuli that we're using. They're drifting gratings moving at different spatial and temporal frequencies. The spatial frequency of a stimulus is the width of one of these bars. It is measured in cycles per degree. The temporal frequency is the rate at which a cycle passes through a particular position in space, and is measured in cycles per second. And importantly, the ratio of temporal frequency to spatial frequency is the speed of the stimulus. And so all of those stimuli that lie along this axis are of the same speed, while those stimuli that lie along this axis are of increasing speed. And so this particular neuron responds with increases in fluorescence from the genetically encoded indicator GCAMP, in this case this is GCAMP3, to high temporal frequencies and low spatial frequencies, or fast speeds. And so, and very selectively, and so that you don't see almost any responses to non-preferred speeds. So if we look at the population of neurons in V1, which, as I showed you, individual neurons are very precisely tuned, the population is incredibly diversely tuned. So here, each dot represents a single image neuron and its preferred temporal and spatial frequency. And so you can see that the population covers this entire range of presented spatial and temporal frequencies. And importantly, if we then looked at the representation of these neurons in cortical space, we saw little organization. So here now, each dot is a representation of a neuron's position in cortical space, either in the coronal plane, where this is the pia and the white matter, or in the tangential plane, looking down on the surface of the brain. And each, this preferred speed of the neuron is color-coded according to this map. That we saw very little organization for speed within this, within either dimension. And this is really consistent with what other people have seen in terms of the lack of orientation tuning or tuning for other features within the mouse visual cortex. It generally seems to have a salt and pepper organization. So despite this lack of organization within primary visual cortex, we saw something really different in the higher visual areas. Namely that neurons in PM and AL preferred very different sets of spatial and temporal frequencies. Neurons in PM preferred higher spatial frequencies and lower temporal frequencies, while those in AL preferred higher temporal and lower spatial frequencies. And importantly, they're even better defined along this axis here of speed. So neurons in AL preferred fast speeds, while neurons in PM preferred slow speeds. So to me, this sets up a really interesting specialization within the mouse visual cortex. That neurons in V1 and LM, which I haven't shown you, prefer a very diverse array of speeds, whereas those in AL and PM are much more specialized, where AL prefers fast speeds and PM prefers slow speeds. So to me, this begs the question of how this specialization could arise. And I see two main possibilities. One possibility is through local computations within each of these areas. An alternative is that neurons in V1 could make specific projections to these downstream targets, and therefore that the specialization could be inherited from V1. And so in order to test this second hypothesis, what we did was use the same approach of making GCAMP injections into primary visual cortex, but now we waited for the expression to be trafficked out to the axons of those neurons within the higher visual areas. And these could then be seen as these arborizations, these islands of fluorescence, both in ex vivo tangential sections, as well as through our cranial window if we cover up the bright fluorescence in primary visual cortex. And so then we can target these islands for 2-photon imaging to look at the responses of these axons to the different types of stimuli that we presented. And so this is a movie, an average movie of the responses of axons within area PM to the different spatial and temporal frequency stimuli. And so what you can see is that when a stimulus is presented, you see increases in fluorescence in these axons, which is due to the spiking of cells that are millimeters away back in V1. And importantly, different stimuli evoke activity in different populations of axons. So this is really great. We can now use this approach to ask the question about whether V1 actually does send distinct information to these different target areas by looking at the receptive fields of these axons within the higher areas. And so the way we analyzed this data was first by identifying all of the axons that were active during that movie by making a maximum DF over F image. And so if you zoom in on a portion of this field of view, what you can see is that these axons are actually comprised of strings of hotspots that we think likely correspond to synaptic boutons. And so we can then use just an automated approach to identify all the hotspots in the field of view and measure their responses to each of the different stimuli presented. And so these 4 boutons here map onto these 4 boutons that have different colors up here. And so you can see that these boutons, like the neurons that we measured in primary visual cortex, are very sharply tuned to spatial and temporal frequency. And importantly, boutons that seem to come up from the same axon share the same tuning properties, which you might expect that's consistent with our expectation that these responses are due to the propagation of action potentials down these axons rather than local processing within the boutons themselves. So, and importantly, that boutons on other dendrites can have very different receptive fields depending on their tuning. So we can quantify this data by making these arrays in which each position corresponds to a particular paired spatial and temporal frequency, and the area of the circle corresponds to the amplitude of the response. And so then we can look at the population of bouton responses across a given area and see what its average tuning is. And so this is a set of experiments from a single mouse, so a single injection within primary visual cortex. What we're looking at is the average response of 3 different fields of view collected on 3 different days, one from PM, LM, and AL. And right away, what you can see is that the information that's sent from this homogeneous population or heterogeneous population in V1 sends distinct information to the 3 different targets. And this was true if we looked at the population of thousands of boutons on average, that the boutons in PM generally preferred slow speeds, the boutons in AL generally preferred fast speeds, and those in LM preferred more intermediate speeds. So this average difference in response in bouton tuning within the targets could potentially arise from 2 different mechanisms, or 2 different anatomical mechanisms, really. One possibility is that there's a difference in the fraction of boutons of a given tuning within each area. And alternatively, since these are simply averages of each bouton within the target, there could also be a contribution of the amplitude of the boutons, and so that those ones that prefer a given speed have larger responses and therefore swamp out the others. So it could either be a numbers game or an amplitude game. And so in order to distinguish between these possibilities, we got rid of the contribution of amplitude and simply looked at the distribution of boutons within each area. So here we're looking on the x-axis at the preferred speed of those boutons and looking at the cumulative distribution. And this really shows that the population of boutons in AL prefer faster speeds than the population of boutons in PM. Moreover, if we look at the median speed within individual fields of view, where individual mice are connected with a gray line, we reliably saw higher median speeds within AL than in comparable fields in PM. So this really suggests that, in fact, there is a difference in the fraction of boutons of a given tuning within a target area. Importantly, since we're just looking at a population of boutons within this area, we can't distinguish between other anatomical mechanisms, such as whether the population of neurons within V1 is more likely to project to AL or PM, or whether once they get to AL or PM, they have larger arborizations. But nonetheless, this seems to explain some of the specialization that might occur. And importantly, that the median speed of the boutons within each area match well to the median speed of the neurons in the area. So this square represents the median speed of the neuronal population, which matches well in AL and in PM. And so this is a quick summary of what I've told you about so far. The population of neurons in V1 is heterogeneous and prefers a range of spatial and temporal frequencies and speeds, whereas those in AL and PM are more specialized. And what we found by imaging axons within these target areas is that the information that V1 sends to these areas is in fact distinct, and there seems to be anatomical routing of this information from V1 to the higher visual areas. And moreover, the function of these projections seem to match the function of the higher areas, and therefore could provide some of the specialization that we see of the receptive fields in the targets. However, I want to point out that while the median speeds match here, that the distribution of preferred speeds of boutons and axons are somewhat divergent. So here in the thin lines represent the distribution of cell bodies, where the thick lines represent the distribution of boutons within each area. And I want to point out that the boutons within AL prefer slower speeds than the cell bodies within AL, and that the boutons within PM prefer faster speeds than the cell bodies within PM. So this really suggests that there might be some additional mechanism of refinement, that in addition to there being these specific projections, that there could be local computations within each of these areas that serve to further refine the receptive fields in these targets that mediate the specialization. So I want to give you a little model of some idea of what we think could be happening to refine these inputs. And one possibility is that the refinement could actually be happening at the synapse from V1 to the target. And so it's well known that synapses can have very divergent dynamics independent of the pattern of action potentials that come down the axon. So for instance, high probability of release synapses tend to have very depressing synapses that result in decrementing membrane potential, whereas low probability of release synapses tend to have facilitating synapses that can lead to the summation of membrane potential throughout a train. And if we impose a threshold on these membrane potentials, I think what this shows is that you could then imagine having very different dynamics of postsynaptic spiking responses in response to these different types of facilitating versus depressing synapses, where depressing synapses might act as a low-pass filter and thereby cut off high temporal frequencies, whereas facilitating synapses might act as a high-pass filter, thereby permitting high temporal frequencies. And so one hypothesis is that AL and PM might be distinguished by having different properties of synaptic transmission at the input stage from V1 to the higher visual areas. That AL might have more facilitating synapses, whereas PM might have more depressing synapses. And so in order to get at this question, we decided to take an in vitro approach by actually recording from neurons within the higher visual areas while stimulating their inputs. However, this is a difficult problem because if you stick a stimulation electrode down in these higher visual areas, you're going to activate a whole range of inputs coming from all sources. And we really specifically want to look at the inputs coming from V1. And so this really is a question that requires optogenetics in order to study it. And so we would make viral injections of channelrhodopsin into V1 and allow the virus to traffic out to the higher visual areas. And then we can make whole cell recordings from these puffballs while stimulating those axons to look at the properties of synaptic transmission. However, I want to point out that it's really important if we want to look at the dynamics of synapses across distances and using optogenetic tools, that we really validate whether channelrhodopsin in particular and optogenetic tools in general can be used to probe the dynamics of these projections. For instance, it's known that channelrhodopsin undergoes significant amounts of inactivation. And so this could potentially make it unsuitable. However, people have shown that whole cell recordings and cell-attached recordings from neurons expressing channelrhodopsin that those neurons can be driven to fire at frequencies of 20 to 30 hertz reliably, even for trains of 10 action potentials. However, axons might be a totally different story. They are severed from their cell bodies, and so and generally, especially these ones, are millimeters away from the source of the opsin generation, and therefore might have lower densities of the optogenic proteins and just have smaller surface areas. So this is something we really wanted to test and see if we actually are able to reliably stimulate these cortico-cortical projections. However, this in itself is a problem because there's really not a very good way to directly record from these axons. As I said, the cell bodies are cut off, and so we can't record from them while stimulating over their axon. And recording from individual axons in a slice, while possible, is incredibly painstaking and difficult. And so instead, we decided to take an indirect approach of recording extracellularly from these axon populations. And so here is an image from a slice that we recorded from the fluorescent image of this axon cloud innervating a higher visual area and an extracellular recording electrode in layer 2-3. And when we shine blue light on these axons, we see this compound potential. And I just want to take a moment to talk about all the different types of conductances that are contained in this potential. So first of all, there's the conductance of channelrhodopsin itself, which then activate through depolarization of the presynaptic axons, activates sodium channels to drive action potentials and activation of potassium channels, which then activates voltage-gated calcium channels, drives neurotransmitter release, and activation of postsynaptic AMPA, NMDA, and metabotropic glutamate receptors, which then drives postsynaptic action potentials, as well as a variety of disynaptic interactions and recruitment of additional excitation and inhibition. So this has got a lot of stuff going on, but really what we want to do is be able to isolate this presynaptic action potential, that fiber volley. And so in order to do this, our first approach was to apply AMPA and NMDA receptor antagonists, these ionotropic glutamate receptor blockers. And that gets rid of responses of these AMPA and NMDA receptors, as well as the postsynaptic action potentials and the variety of disynaptic conductances that might be evoked as well. And so you can visualize this field postsynaptic potential, which is composed of all these monosynaptic and disynaptic potentials, as well as probably a lot of voltage-gated conductances. Whereas the remainder of this is going to be mostly presynaptic conductances, as well as maybe some activation of metabotropic glutamate receptors. So then to further isolate that presynaptic action potential, we're now going to apply cadmium to block calcium channels. And so this is important because we don't want the calcium channel conductance to contaminate our sodium channel or our action potential response, but it also serves to block synaptic transmission, thereby blocking metabotropic glutamate receptors as well. And then now we can finally apply tetrodotoxin to block voltage-gated sodium channels, which has the added effect of blocking action potentials, as well as a lot of the voltage-gated potassium conductance, and leaving us with this red trace, which likely corresponds to the opsin potential itself. And the subtraction of these will give us our fiber volley. And so we contend that this fiber volley is going to be a good measure of the average activation of these presynaptic axons from V1 for a couple of reasons. One of which is as we increase the light power of the LED, we see an increment in both the amplitude of the opsin potential as well as the amplitude of the fiber volley itself. And so this is giving us now an average response of the population of axons that we're activating coming into this higher visual area. And I would actually even argue that this is, while indirect, maybe a better measure than recording from individual axons, because it gives us an unbiased approach to look at the entire population of axons that's recruited. And moreover, it's going to be the identical configuration to what we'll be looking at ultimately when we're doing whole cell recordings from these axons, or from these neurons and stimulating these axons. Okay, so what happens now if we stimulate these axons at high frequency? So this is, here I'm overlaying fiber volley responses. This is channel rhodopsin stimulation at 20 hertz, where the black trace is in response to the first pulse, and increasing lightness corresponds to later pulses in the train. And what you can see immediately is that the fiber volley becomes much smaller and slower. And this is true if we looked across a range of different temporal frequencies, that even as slow as 10 hertz, the axons were totally unable to follow stimulation with channel rhodopsin. Importantly, that not only did we see changes in the reliability, but we also saw changes in the fidelity, that the timing of the fiber volleys became later and later during the train, increasing up to 50% in their latency. And importantly, this is not simply an intrinsic property of these axons. If we've now placed an extracellular stimulating electrode in the tissue and looked at the recruitment of fiber volleys, we saw that we could very well stimulate axons in the cortex. That they had very high reliability and stable latency. And so I just really want to point out here that this really suggests that channel rhodopsin is totally unsuitable for stimulation at high frequencies, or even my guess is at frequencies well below 10 hertz. That instead we need to look to other tools for this approach. And in order to start thinking about what tools we might want to use, we wanted to try to really understand, what's causing this effect. And our intuition is that the high light powers that we need to use to evoke action potentials in these axons really exacerbate the inactivation of these channels. And so here now we're looking at the opsin potential that was recorded. So this is now in the presence of all of our antagonists. And again, where we've overlaid at 20 hertz these these potentials in black is the first pulse and gray is the last pulse, that we see this marked use-dependent decrease in the amplitude of the opsin potential that we think is probably due to channel inactivation. We don't think this is due to changes in driving force, because if we do whole cell recordings from neurons, we see strong inactivation of these currents as well. So this leads us to think that we really need to use new tools that maybe undergo less inactivation. And that brought us to two newer opsins, OChief and Kronos. OChief is developed in Roger Chen's lab, and Kronos here by Boyden. And both of these opsins, so these are whole cell recordings now looking at whole cell currents normalized, that OChief undergoes very little inactivation up to at 100 hertz, you start to see some fall off. And Kronos is also quite good. It's much better than channel rhodopsin, although it starts falling off maybe at 40 hertz or so. So now we wanted to test whether these newer opsins might be better tools for activating these axons. So we went back to stimulating the axons themselves and recording the fiber volleys. And so here are just two example experiments, one from OChief and one from Kronos, stimulating at 20 hertz. And you can see already that these fiber volleys are much more reliable and stable throughout the train. So this is a summary of data, again, for OChief and Kronos, that OChief is remarkably stable up to 60 hertz, starts to fall off at 100 hertz. And Kronos is very stable up to 20 hertz, falls off a little bit at 40, and a little bit more at 60. Importantly, they also show relatively stable latencies, maybe not as stable as the electrical microstimulation that I showed you, which showed some supernormality for people that were noticing. But that, again, it's fairly good, and you really, by eye, can see very little change in the timing and the amplitude of these fiber volleys. So it seems now that we found a couple of tools that we can use to reliably stimulate axons in vitro and look at the properties of synapses. And so now, returning back to our preparation, where we have these axonal arborizations that are innervate AL and PM, we can now make recordings from these areas to look at the properties. And so just as a first pass, I want to show you some data actually looking at the field potentials themselves. And so this is a field potential recorded in PM. As this is, again, I've been showing you mostly the opsin potentials and the fiber volleys. Now we're going back to this initial condition where we just have the control minus the AMPA and NMDA receptor blockers. And importantly, we're really just going to isolate this rising phase, because that's the part that we really trust as being probably the monosynaptic component of this EPA. And so what you can see is that at lower frequencies, there's actually very little dynamic of the input from V1 to PM. But then as you increase frequencies, you actually see something that's very commonly observed, which is, this is facilitation followed by depression. And indeed, that's what we saw across the population in PM, which was actually distinct from AL. So whereas at lower temporal frequencies, both of them, inputs to AL and PM were relatively stable, showed very little plasticity. As you increase frequencies, they began to diverge, whereas inputs to AL become more facilitating and inputs to PM undergo a bit more depression. And we don't think this is simply due to activation over the axon terminals themselves that were causing some kind of pathological calcium influx. Because if we move our laser down to layer 5 and activate there instead, we see a very similar response. So this was consistent with our model, this idea that maybe the inputs from V1 to the higher visual areas might be specialized, where those to AL might be more facilitating than PM, although maybe not as dramatically as I schematized. We're currently doing whole cell recordings, trying to replicate this to really look to see whether it is those monosynaptic EPSCs that are distinctly modulated in temporal, by different temporal frequencies. But we're also continuing to look for other mechanisms that might support the specialization within these downstream areas. So for instance, where excitation might mediate this different filtering properties, you can imagine that a complementary inhibitory circuit could further refine these inputs. So for instance, if you added a facilitating inhibition to a depressing excitation, you might further emphasize your low-pass filter, whereas a depressing inhibition plus a facilitating excitation would further emphasize your high-pass filter. And so we know that there are different subtypes of inhibitory neurons that act exactly in this way. That, for instance, parvalbumin expressing inhibitory neurons receive strongly depressing synapses and therefore impinge on pyramidal cells with depressing inhibition, whereas somatostatin positive interneurons tend to receive facilitating synapses and therefore impinge on their target pyramidal cells with facilitating inhibition. And so so one hypothesis is that, for instance, there might be different densities of parvalbumin positive and somatostatin positive neurons across different cortical areas that might match with this model to further refine these different temporal frequencies. And so the way that we approached this was by using 3 different lines of transgenic mice. We used somatostatin iris Cree that we crossed to flex TD tomato, parvalbumin iris Cree that we crossed to flex TD tomato as well, and then these GIN GFP mice, which is a transgenic line that expresses GFP specifically in a subset of somatostatin cells that are likely Martinati cells and are the ones that really receive strongly facilitating excitation. And then what we did is made viral injections into primary visual cortex to express GFP in these 2 lines, or TD tomato in this line, to be able to identify the axons of the higher visual areas, axons innervating the higher visual areas, to essentially define our regions of interest so that we were comparing retinotopically matched regions within the higher visual areas. And so this is an example section. So we made 70 micron sections through the brain. And then used confocal imaging to create these stacks. And so in magenta here is the TD tomato expression of the somatostatin expressing interneurons in the SOM-Cree line. And you see the expected bright fluorescence in layer 1, that a lot of these neurons send their axons up to that layer. And in cyan, we see the V1 axons innervating the higher visual area. And so we really use this to define our region of interest and to maintain constancy of representation. And importantly, this is an example of a DAPI stain from this slice that allows us to identify the laminar location of these neurons. And so then we just went through and counted, or one of the undergraduate students went through and counted all of these inhibitory neurons and identified their location along the coronal axis of these slices. And then we repeated the same thing for GIN cells as well as the PV Cree cells. And so I just want to point out 2 things. One is that the GIN cells generally tended to be in the more superficial layers, and so maybe correspond more to the somatostatin cells in the superficial layers of these mice. And secondly, that the inputs from V1 to the higher visual areas tend to arborize mostly in layers 2, 3 through 5. And so we really restricted our analysis to those layers specifically, since that's the input that we're thinking is going to be modulated. So these are the results of a lot of cell counting. So in the PV Cree line, for each of these lines, what we measured is the relative change in density in AL versus PM. And so here we saw a 20% increase in the number of PV positive interneurons in AL compared to PM, and a 15% decrease in the number of GIN cells in AL compared to PM. Interestingly, if we looked at the SOM-Cree line, that it came out around equal. But if we then divided these neurons up according to being layer 2, 3 versus layer 5, now these segregated kind of interestingly, where the SOM-Cree cells in layer 2, 3 look a little bit more like the GIN cells, as I pointed out, kind of anatomically, whereas the SOM-Cree cells in layer 5 are actually more strongly represented in AL than PM. So this really suggests that there may be some kind of difference in the recruitment of inhibition to these different areas that could support the specialization in the temporal frequency tuning that we observe. And we're currently doing whole cell recordings to look at this as well, to look at the recruitment of inhibition and the strength of inputs as well onto each of these different cell types within these targets. But either way, I was actually really surprised by the result that there are these different densities of inhibitory neurons across areas, and really suggests that while there might be canonical microcircuits within cortex, that the balance of those circuits might really be specialized across different areas, and it's probably going to be interesting for understanding how different modalities are processed as well. And so in summary, I'd like to argue that in addition to having shown you that there are specific projections from V1 to the higher visual areas, that there also seem to be local computations within these areas. That there are potentially differences in the inputs from V1, the V1 synapses to their targets, as well as the recruitment of local inhibitory circuits, and that these may be specialized to refine the tuning and of the neurons within these areas. And so I'd just like to end by thanking a lot of people. First, Jeff Sims was an undergraduate who got all of the behavior working that I showed you at the start. Mark Anderman and Vincent Bonin and Clay Reed in the Reed Lab. I did all of the imaging experiments with them. John Mansell and Mark Heisted were instrumental in getting a lot of those behavioral experiments going as well. Charlie Haas did all of the slice electrophysiology, and with Amy Crystal did the anatomy. And Greg Horowitz provided us with some viruses. And I would like to thank my funding and all of you.